Greetings, everyone, and thank you for joining the webinar today. And I wish everybody a good morning, a good afternoon, or good evening to you, wherever you may be. And today's webinar is sponsored by Logan Instruments, and the topic of today's webinar is de-risking your bioequivalence studies, or best practices for increasing IVIVC of oral dosage forms. We're joined by two very distinguished speakers today. But before we get started, I'll go through a few housekeeping items, if you will. This webinar is scheduled for 1 hour. We will do our best to maintain to that schedule. That includes some question and answer session at the end. This webinar is being recorded, and it will be posted to various social media, websites, YouTube channels, etc. So if you're not able to attend the full webinar, we can get you a video of it later on. At any time during today's webinar, if you have a question for one of our speakers, you'll see a small Q&A button at the bottom of your screen. If you just press that, you can type in your question, and we will get to the Q&A at the very end of the webinar. And if we don't get a chance to answer all of the questions in this session, we will do our best to have the speakers follow up with you via email. So without further ado, let's proceed to presentation number 1. And our first speaker is Professor Massimiliano Pollo di Cagno. And his topic is in vitro permeability testing, or increasing pharmaceutical success rates while promoting sustainability. Professor Di Cagno is a associate professor of biopharmaceutics at the University of Trieste in Italy. He has a PhD in pharmaceutics from the University of Southern Denmark. His expertise is in many areas, but specifically in permeability, passive diffusion of drug, and physical pharmacy. He's the author of 32 peer-reviewed articles, including a book chapter, and he holds 2 patents. So without further ado, I think we'll turn it over to Professor Di Cagno, and good afternoon to you, sir.
Thanks a lot. Thank you, Keith, for a nice introduction. I hope you can hear me well as the audience.
Yes, perfectly. Thank you.
Then I will just share my screen, and I will then start the presentation.
Great.
Okay. I hope that everybody can see my screen. Okay.
Yes, looks very good. Thank you.
Okay. But then I will just start. First of all, thank you, Keith, for inviting me, and Logan to invite me in this nice webinar where we are going to discuss a very relevant topic for my research and my interest. So the title of my presentation is In Vitro Permeability Testing, Increasing Pharmaceutical Success Rate while Promoting Sustainability. In this presentation, I will just sorry, I cannot move. Okay. During my presentation, I will introduce the issue of sustainability in early stage drug development. I will present some of the most common permeation setups that are available nowadays, and I will then focus on the PermePad GAT, the artificial biomimetic barrier, which I believe is the game changer in terms of permeability studies. And then I will leave the word to Professor Lin, which will go a step forward and discuss more advanced dissolution permeation setups and case study, and results that has been obtained utilizing the PermePad barrier. So first of all, the gamble of drug development. I think most of you knows that developing a drug from A to Z, statistics are very variable, but we are talking in the range between 1.2 to 1.3 billion of euros and at least 8 to 10 years of development. When it comes to generic drugs, this might be lower indeed, but anyway, if we're talking about a complex generic drug, the cost can easily rise up to 20 million euro, and the time of development shifting from 2 to 5 years. So these topics is extremely relevant also for generic drug development. And why the cost of drug development are so high? Well, there are different reasons, but definitely when it comes to early stage drug development, so formulation testing, we have the issue of the unsustainability of animal testing. Animal testing is still a practice that is heavily used both in Europe and all around the world to validate formulations and find the best candidate to move forward to in vivo testing in humans. So the reason that the animal testing is not decreasing over the years, so even though the European Union made a lot of effort in this direction. So the last statistics give us number of between 10 to 11 million of animal sacrifices every year, only in Europe. And this is just reported data. But these animal testing are very costly. If we count direct and indirect cost of animal testing, if we have a set of 10 animals, this can easily go up to $50,000, producing an amount of 200 kilograms of carbon dioxide, and minimum time for this experiment to have the results is at least 2 weeks. So definitely this type of experiment are not sustainable, and not only unsustainable, most of them often they're not reliable, because sometimes in vivo animals do not match in vivo humans. And to try to find a solution to that, already in the early 2000, a brilliant mind in our field, starting from Amidon, introducing the BCS classification system, going to Kenzie and Pfizer in '98, where the first biomimetic barrier, artificial biomimetic barrier, the PAMPA, the parallel artificial membrane permeability assay, has been introduced. Going through Lipinski, that in 2001 introduced the so-called Rule of 5, gave the momentum to the concept of permeability, or better, upper impermeability. And I will come back later to the concept, but how do we measure permeability? Permeability is measured utilizing a barrier which have to be crossed by the chemical entities we are studying, and this barrier can be cellular based. For example, the very famous CACO2 model introduced by Artuson back in the 90s. But the EpiSkin, which is actually recreating the skin, a cellular-based model for studying skin permeation of drug and chemical entities, or the most common ex vivo tissue, so where actually the tissue is taken from animals, even humans. In this case, I reported the picture of a ear of the pig, which is actually a very common model for example, skin studies. But several cell-free biomimetic barriers has been developed. One, the very basic one, are better to call them membrane. For example, regenerated cellulose, trans-hem for use for skin permeation studies, but these barriers definitely are not biomimetic. They somehow impose a resistance to the transition to the drug, which is often not biomimetic, because they do not have any biomimetic component inside the barrier. As I named it before, the PAMPA, the parallel artificial membrane permeability assay, this is definitely the state of the art, or it has been the state of the art for in vitro artificial barrier permeation studies. This is basically a filter which is soaked in organic solvent where the lipids are dissolved. And then the PermePet GIT, which has been introduced in the market around 2016, and which is actually gaining a lot of interest nowadays. All this barrier works with the same conceptual model that can be represented by the diagram of the figure or the graph in the left side of the screen. So we have a donor compartment here, where we place our drug in solution. Then we have the barrier, one of the type I presented before, and then we have an acceptor compartment. So making a series of assumptions, what we do is to try to measure the flux, so the amount of drug that is actually permeating through the surface area over the time. The flux cannot be compared between different drugs because the flux is concentration dependent, is related to the gradient of concentration. So that's why it has been more common to utilize the so-called upper impermeability coefficient. The upper impermeability coefficient, as you can see, is a velocity. Centimeter of a second is the normal way to express. And this is the flux that we measure in this experiment, normalized over the concentration of the drug in the donor. In this way, we find a parameter which will actually tell us directly the permeability, so how fast the drug or the chemical entities under development will permeate, giving a reference compounds for permeation. And the experimental setup can be done by sampling over a regular time point, obtaining a nice linear regression, or as a more practical and I would say industrial approach is just to incubate, for example, our system for some hours and just get the so-called endpoint slope by measuring only 1 point. Of course, we lose a lot of understanding of what is happening in the first phase, but it is definitely useful for high throughput screening. Which are the setup that we can utilize? Well, the diffusion cells, that are the cells that we utilize for such experiments, are of different type. We have the vertical diffusion cells, the horizontal diffusion cells, and nowadays we have the multi-well diffusion cells. For example, PermeaPad offer the multi-well plates where every experiment will be a barrier, so you can really have a nice high throughput screening of this. When it comes to classical vertical diffusion cells, Logan Instrument has very advanced device and offers very advanced devices like the one you see here, where the experiment is basically completely automatized in terms of setting up, sampling, and washing. So at the end of the day, you just you get a plate with all the samples that just need to be analyzed with a suitable analytical method, UV, HPLC, mass, whatever it is. Where PermeaPad GIT stands with the golden standard, the pump, well, indeed we have PermeaPad shows over the years to have a lot of advantages, especially in terms of stability, resistance to excipients, so suitability for formulation testing. But what I believe is also very, very interesting is its highest biomimetic properties, because PermeaPad GIT can account for both transcellular and paracellular transport, which are both extremely relevant physical mechanisms when we have the passive transport. And why this is happening? Because this is a side, like the size, a drawing of the size of the PermeaPad. Once the PermeaPad, it comes in pod, but it also comes, as I said, in multi-well plates, get hydrated, it spontaneously forms a liposomal gel inside 2 layers of inert material, which is represented by the liposomes I draw here. In this case, the drugs then can do 2 things. It can permeate through the cells, so the transcellular transfer, but it can also go through the interspaces between liposomes, mimicking the paracellular transport. And this has been proved in 2022 by Ericsson and the group of Martin Brandl and Annette Brandl at SDU, where they actually have play around with tonicity and try to shrink and swallow the liposomes that are comprising the liposomes, and they actually shows that actually by changing the tonicity of the acceptor and donor compartment, they could achieve a modulation of the permeation, given by the fact that they could actually open and close the junction between the liposomes. So when it comes then to biomimetic and in comparison to what is available, like the Pampa, for example, Pampas offer somehow to mimic small hydrophilic compounds because they are the one that generally tends to go through the paracellular pathways, for example, the caffeine. In this Pampa GIT matter, in the Pampa GI that has been reported by this bioassays company, the caffeine has been ranked as a medium-low permeable compound. But in reality, we all know that caffeine is one of the most bioavailable compounds, which reaches 100% bioavailability very fast after oral administration. And what PermeaPad does is actually to rank it accordingly. And in the PermeaPad setup, the caffeine is definitely the most permeable compounds we often find. This is a presentation of how Amidon proceeded in 1995, the comparison between the upper permeability and the fraction of those absorbed in human. So that was the goal to achieve, like with the compounds, and actually with a set of 13 compounds tested back in 2020 from Jacobsen et al. They were able to actually show a very nice correlation between the PED permeability and the human fraction absorbed. More recently, Butner also et al. In 2023 tested the much higher set of compounds, 50 compounds, and they were actually, and they tested utilizing mass spectrometer, and they actually were able to rank these compounds between a range of high permeable and low permeable compounds. And that correlation was very much in accordance with the available in vivo data for such compounds. As I said, the PermeaPad can actually be utilized for enabling formulation testing. And now I will show you an example. This is taken from the work of Kit Gord from 2021, and this was achieved at the University of Copenhagen. In this case, they were interested in testing sneeze formulation, which drug release was associated to the digestion of these sneeze systems. So they have incubated the sneeze with biomimetic relevant media, which also contain lipophilic enzymes. And after incubation, they transferred the supernatant or this mixture inside front cells, which were equipped with the PermeaPad GIT. And then they started to measure the permeation of the API in the septal compartment. And what they find out, they were testing different types of formulation, and they obtain, as you can see, different type of cumulative permeated amount. So the flux profile of the formulation they tested were very different according to the formulation utilized. But what is more remarkable is that they compared the area under the curve obtained in vitro, so the amount or the complete amount of drug that was permeated over the 3 hours period, and they compare with the available in vivo data in animal model they have in the house. And what they found out was a very nice correlation, almost linear correlation, 0.92, between the formulation, which in the in vitro data they obtained with PermeaPad and the in vivo data they had in the house. They concluded that PermeaPad was definitely a very nice and reliable tool to investigate in a rapid and cost-effective way these types of enabling formulations. If we want to go a step further, then we have to introduce the concept of dissolution and permeation. That means that instead of having just a dispersed formulation, we actually tested our original solid dosage forms. In this case, I represented tablets, which find itself inside the dissolution standard vessel. And with an insert, it is possible to also quantify or estimate the amount of drug that can permeate through an artificial barrier inside an hypothetical septal compartment. So with this type of setups, we can assess both the dissolution rate, if we can measure the amount of drug that is dissolving over time, but most importantly, we can also have a good estimation how much will permeate physically inside would be actually absorbed. One of the systems that can actually allow this kind of study is called the permeation, and this is also another instrument that is provided by Logan Instruments. And the speaker that is going to follow me, that will speak after me, will actually show some case studies of dissolution permeator testing. Before concluding my speech, I would just like to suggest, if you are interested in such topics, to have a look at this review from 2023, which was written by me and co-workers, where we have tried to summarize all the available, commercially available in vitro artificial systems for permeability studies, which are available from classical permeation studies, but also going into the dissolution and permeation studies. And with this, I would like to conclude. Thanking Keith again and Logan Instruments for inviting me, PermePad and Fabio for the support that they daily give me in my research. And if you are interested in collaborating with us, we are Trieste, and we are very happy to give you support in this innovation and sustainability through pharmaceutics.
Thank you very much, Professor De Canio. That was very interesting. So you've covered the barrier, the biomimetic barrier, and your data is very interesting. And I think we'll probably have some good questions for you at the end today. And with that, maybe we'll turn it over now to our next presenter. Hold one moment here. Our next speaker is Dr. Yan Lin, and she's going to speak today on the application of Logan dissolution devices and PermeaPad, PermeaPad being the biomimetic barrier that Professor De Canio just presented. Dr. Lin is an instructor at China Pharmaceutical University, School of Basic Medicine and Clinical Pharmacy, and she was educated at the Ohio State University College of Pharmacy, which is here in the United States. And she specializes in a research field right now, individualized and translational medicine under the integration of clinical pharmacy, clinical pharmacology, and modeling simulation. So with that, Dr. Lin, thank you very much for joining us today. It's evening time there in China, and we'll turn it over to you.
All right, thank you. So can you all see my slides?
Yep, we see your PowerPoint program. Yes.
All right, thank you.
Oh, there we go.
All right, good evening and good morning, everyone. Welcome to today's webinar, and I am Yan Lin from China Pharmaceutical University. And today I will be guiding you through how Logan's permeable and PermeaPad can be a game changer in lowering bioequivalence risk for pharmaceutical company. And as we all know, bioequivalence is a critical aspect of drug development, especially for generic drugs. And the failure in BE tests can lead to significant financial losses and delays. And that's where Logan permeable and PermeaPad come into play. So this innovative tool can help us better understand and predict drug absorption, which is the key to minimize BE risk. So our research will rely on biopharmaceutical evaluation platform. So this platform really integrates multiple attributes of API and formulation, and also along with disease requirements, drug mechanism, and human physiological information. So it will serve as a bridge between drug manufacturing and clinical outcomes, aiding in prescription design, stable production, post-market changes, and research and development of improved high-end preparations. So when it comes to orally administered drugs, the absorption really is complex. It involves disintegration, dissolution, memory transport, and a gastric empty and a transit. So among them, permeability is a key parameter in the absorption process, which represents the extent and the rate of memory transport for a drug. So the in vivo dissolution devices we traditionally use cannot fully replicate the in vivo environment. So this is where bio-relevant dissolution media and complex dissolution devices such as permeable step in to stimulate real-life conditions more accurately. And another useful tool to help us connect between formulation characteristics and absorption is PBBM. So PBBM stands for physiologically based biopharmaceutics modeling. So it focuses on the relationship between drug formulation characteristics and absorption, and can be used in formulation development to evaluate the dissolution and absorption characteristics of different formulations and predict the bioavailability of drugs. And in BE tests, it can be used to assess the bioequivalence of drug products and even provide a basis for BE exemption in some circumstances. It can also be used to study the food effects for a certain drug and the influence of dosage from changes on drug absorption, and etc. And the following are two cases where the multiple methods mentioned above are applied to reduce the BE risk. And our first case is screening precipitation inhibitors for genfetinib, which is a weekly basic drug of BCS2. So BCS2 class, they are highly soluble in stomach acidic environment, however, faces a significant drop in solubility in the small intestine environment, which is neutral to alkaline. And this can lead to drug precipitation and a reduced absorption. And given that about 75% of marketed drugs are weak bases or their salts, so finding the right precipitation inhibitor is crucial for generic drug prescription development. So we choose genfetinib as an example. So genfetinib is BCS2 class weakly alkaline, and its solubility is really pH dependent. So the reference preparation is the original drug, Iressa from AstraZeneca, and the testing preparation is the generic drug from a pharmaceutical company. So the bottom is the pre-experiment BE study results. And as we can see, the test preparation showed a Cmax BE ratio of 85.11%, with 90% confidence interval of 73.98 to 97.91. And similar non-BE results were also observed for AUC. So these results really highlight the high risk of non-BE for genfetinib. And to address this, we developed and verified a dissolution method with the in vivo predictive power. And by combining with PBBM, we were able to screen appropriate precipitation inhibitor for genfetinib. So for two-stage dissolution test or two-stage dissolution experiment, we use biological dissolution media, both for gastric and a small intestine solution, stimulation solution. And the pedal method is employed. And the two dissolution cups were connected by the peristaltic pumps to stimulate the movement process from the stomach to the intestine. And a different volume of intestinal fluid, different gastric empty rate, and different propeller speed was tested to better stimulate the real human body environment. And the release parameter and the sedimentation parameter were evaluated for the results. So with two-stage dissolution method and PBBM, we finally determined the optimal experimental conditions for further study. And then we examined the effect of different polymers as well as their dosage on the drug supersaturation and precipitation in vitro. And through this predictive dissolution method guided by PBPM, we finally identified polymers with precipitation inhibition potential. And our second case is model-informed prediction of food effect for oral drug A. Since this research is just under publication, so we will use drug A as abbreviation. So as we all know, food can dramatically alter the physiological environment of GI tract, including prolonged GI empty timing, elevated pH, so increased pH of the stomach environment, increased secretion of bile acid. So the core of food effect prediction really lies in accurately assessing the FA. So FA stands for fraction absorbed, and really assessing FA of the drug in both fasting and eating conditions, since FA directly determine the differences in BA in bioavailability and bioequivalence. So we developed a dissolution permeation system. So in our system, we study the solubility of drug A in a biorelevant solution medium, including fasted and fat states. And the donor pool was set up with a USB-2 device using passive and FASIV media. And the receiver pool featured PermePad as the permeability memory and a plasmamimic ASB solution as media. So based on the dissolution permeation experiment results, we conducted an IVIVR evaluation. So we used the tool drugs to verify our method, establishing the relationship between the permitted amount and FA using this tool drug. So this is our results. So unlike the traditional method that established relationship between PAPP and FA, so our method established the relationship between FA and the clinical dosage. And this approach allowed us to predict the food effect on drug A's absorption with greater accuracy. So like I mentioned in case 1 and case 2, so Logan's Permetrol and PermePad offers a robust solution for reducing B test or reducing B risk by providing valuable insights into the drug absorption characteristics. And by leveraging multiple methods like Biopharmaceutics evaluation platform, PBBM, the solution permeation system, we can make more informed decisions during the drug development process. And that's all from me. And thank you for joining me today. And I'm happy to take any questions you may have. Thank you.
Thank you very much, Dr. Lin. That was great. So we've now established that with a biomimetic membrane or barrier and a suitable device to evaluate the dissolution medium as it permeates through that biomimetic barrier, that we can have some data where you can compare the generic formulation with the innovator formulation with respect to GIT permeability in a in vitro model. So we thank you for that. And in fact, I want to thank both speakers. And before we get to the Q&A section here, maybe it will give a little more some insights, if you will, into the specific instrument that we're talking about here. And we call this instrument Permetro. Permetro is the device that you see on the far right of your screen here. Make sure I've got my laser pointer on. And Permetro is an interesting device. This is a patented Logan device. And inside of Permetro, you can see we have 8 or so side-by-side or flow-through cells. And if you take a cross-section of these flow-through cells, you'll see here that we've got 2 sides separated by a PermeaPad or the biomimetic membrane here. And on the right side, we call this the dissolution chamber. And in this dissolution chamber is where the pumping occurs from your dissolution vessel online via your Logan dissolution tester. And that flow rate can be controlled and fully programmed. And on the left side, what you see here is the receptor chamber. And these are offered in two different volumes, 5 ml receptor volume or 12 ml receptor volume. In this chamber, you've got a mesh screen sandwich, which holds the PermeaPad GIT biomimetic membrane. At the bottom of the cell, we've got the mixer. So at the prescribed aliquot timing points, you can program where the pumping will occur from the bottom of the receptor media inlet, and you will pump your receptor chamber media all the way out into your collection vial. So this is a cross section of the setup that we have in the Permetro system. Now this Permetro system, as you can see here, is online, and this system works with our 8-vessel tester, which is what's pictured here. And here's another cross section of how the pumping fluidics will work. So your dissolution solution is pulled through the auto-sampling pump system, pushed from there, from the precision syringes into the Permetro cell in the dissolution chamber. And then typically it's recirculated back into the vessel. Now there are different setups where you can also collect that dissolution media or solution, but in this particular example, we're doing a closed loop pumping. Then simultaneously, at the prescribed aliquot time points, you can pump your receptor solution through that Permetro cell directly into the vials or test tubes that you select. So what you have here is really sort of an online tool that allows you to take the dissolution profile and match it up with the permeation through that PermeaPad GIT barrier. Now the Permetro system is compatible with the Logan apparatus 1 and 2 dissolution setups, and those come in 8-vessel or 12-vessel systems. This can also be used online with the Logan apparatus 3 system that we call Disso 3, and it can even work with the Logan apparatus 4 or the Super 4 system from Logan. So you see there's a lot of flexibility, a lot of compatibility depending on the permeation study that you're selecting. And we've got a lot of really good questions here I think we'll get to in a moment. As far as Logan is concerned, if you haven't heard about Logan before, we've got a broad portfolio of testing instruments that span from all the different USP apparatus types. Apparatus 1, 2, 3, 4, 5, 6, and 7. We offer a mobile media degasser system, a clean in-place vessel washer, of course the Permetro and PermeaPad products. We have a whole portfolio of diffusion cell equipment, whether it's dry heat, water jacket, manual systems, automated systems for IVPT, and of course for IVRT. The IVPT system has the full media replacement, and we have many more products as well. So you see it's a pretty broad portfolio, but we see a lot of emphasis today on the bioequivalence and the permeation area of the generic formulations. And using the Permetro product, it really allows you to compare, of course, using the biomimetic membrane, you can compare the innovator formulation and its permeation characteristics. You can compare that with your generic formulation and its permeation. This is not a substitute for in vivo studies, but it certainly gives you the ability to de-risk your in vivo studies. And with that, I think we will, one more slide here, one more about Logan. We're based in New Jersey. This is our global headquarters here in the USA, where we have all of our R&D applications, manufacturing, et cetera. And we have 2 other locations located in Shanghai, China. One is in Shanghai, one is just outside of Shanghai. We have also co-R&D facility there. We have manufacturing application support. This global footprint really gives Logan the unique ability to manufacture products in the USA and in China so that we can go to the market with competitive price points depending on the customer's budget needs. With that, maybe I'll ask our presenters to unmute themselves and come back online. I see we have a number of questions here. We may not have time to get to all of them, but if you'll allow me, doctors, I will maybe ask one or two questions first, and then we'll let you kind of go through and answer some as time is permitted.
Yes.
Okay, great. So maybe we will ask are you able to see my slides?
Yes, I can.
Okay, good. I'm seeing a question that says you can't. Maybe the first question we'll ask Professor Di Canio is, here's a good question. What's the difference between PermeaPad and PermeaPad GIT?
It's just a semantic. I think that the terms GIT has been introduced later on as the PermeaPad technology is developing through mimicking different types of barrier. They decided to discriminate the PermeaPad GIT, so the one that is designed to mimic the GIT and other type of barrier that are under development nowadays. For example, the PermeaPad skin or other type of barrier. So it's just semantic.
Excellent, thank you. And Dr. Lin, what will be the calculation procedure that will help predict that the product is going to pass the upcoming BE study?
Actually, there are some standard equations you can calculate the permeability. So as shown by Dr. Di Canio, like PAPP, we have PAPP, we have PEFF, and there are some well-recognized equation by the industry to calculate, how much drug kind being permitted through the memory.
Very good, thank you. And sticking with Dr. Lin here, I'm going to read the question as is. Food effect identification, maybe does it have any comparative data with a biological system?
Yeah, definitely there are a lot of publications that have some comparative data like to compare the in vitro and in vivo results. However, for our research, unfortunately, we don't have the comparative data regarding to the biological results. However, there are other like publication involving other drugs that definitely have comparative data. Yeah, there are tons of them.
Very good, thank you. Dr. Di Canio, here's one maybe you can help with. How is this model useful for extended release formulations?
Well, it is. I think that this question can go through both me and Dr. Lin.
Okay.
I think it's is referring to the fact that if you have a control release formulation, most likely a solid dosage form, how can you test this in the solution permeation studies? You can, if you utilize a permeation system or the solution permeation system, which is online, it is exactly the same if you test a fast release or a delayed release. You would just get a different profile in terms of the solution and in terms of absorption, which hopefully will mimic better the in vivo condition. And then the results should be much more aligned with what you see in vivo.
Dr. Lin, do you have anything to add to that?
Yeah, actually, when we build up PBBM, so this model, actually, we need a lot of parameters. So for example, we need drug-dependent parameters, we need formulation-dependent parameters, and then we need physiological parameters. So when you build up the model, so if that is an ER formulation, we can adjust the according parameter to mimic this scenario. So for example, we can adjust the drug-dependent parameters. For example, the absorption process may be delayed based on the like log P, or the formulation dependent parameters. So we can adjust the according parameter to really mimic certain scenario. So actually, and also when we develop dissolution and a permeation like experiment, we can also adjust a certain step to really mimic the ER formulation. So that's what I can think of. Yeah.
Excellent. Thank you very much. We have several questions here. We're going to try to get to as many as we can. Maybe we'll ask both speakers and we'll let you arm wrestle on who wants to answer. How do you maintain sink condition in the permeation chamber with very low volume and regular sampling?
Maybe I can take this one first,
Okay.
because I think he's referring to maybe multiple plate cells where you have very small volume. Well, in sink condition is always a parameter that is considered to be crucial. Indeed, it is important. What you can do, the easiest way is to sample out a significant amount of the acceptor compartment and replace it with a fresh buffer or PBS of the condition you're using for mimicking the acceptor compartment. Another way is just to utilize a different mathematical approach to feed the data that will take into account the fact that you are not in sink condition, like considering the austere water layer, for example. So this is just a matter of how do we interpret data. If you want a standard approach for interpreted, like the one I presented, permeability, flux, etc., then you need to have a linear regression. You need to come across the steady state. But there are other ways that can be utilized, other mathematical approaches that can feed the data and give you the same results, even though in non-steady state condition.
Excellent. And maybe this one's for Dr. Lin. What is the calculation factor in, I think, permetro and normal dissolution correlation? I'm not sure I'm understanding that question. What is the calculation factor in permetro and normal dissolution correlation?
I don't really understand what's the real question.
Yeah, maybe if that question just can be rephrased, we'll take a stab at it. So thank you. Let's see, Professor DeCagno, how is it different, a PermeaPad from PVDF membrane with and a lipid solution?
I assume this is referring probably to a biomimetic barrier where you have a filter which is soaked in organic solvents and lipids, like more like the Pampa, I presume. The difference is the physical method of permeation that the drug will do. In the case of this lipophilic barrier, like the Pampa, you just have like a partitioning effect. So the drug will change from a water phase to a lipophilic phase, and then water crossing to interfaces, physical interface. In the permeapad, you have this, but the drug goes through liposomes, which are much more closer to cell bilayers in terms of structure. Plus, you also have the paracellular path that is this water channel between the liposomes. So that it's much more physiological, the mechanism of permeation when you have permeapad than we have all the other methods.
Thank you.
You're welcome.
Okay, Dr. Lin, this is a series of 3 questions it looks like here, and we'll kind of take them one at a time. Is there any factor required to simulate the permetro data with the human GIT system? And I think you touched on it, but maybe you can elaborate. Any factor required to simulate the permetro data with the human GIT system?
Yeah, actually, like when we build the PBBM model, we need to collect the physiological parameters. So for example, if we want to simulate the absorption for certain drug in human body, so we will collect the physiological parameters for humans. However, for example, for the preclinical phases, if we want to simulate the absorption on rats, so we will collect the physiological, some physiological factors or parameters on rats. So for example, the pH, the intestinal fluid, the volume of the gastric fluid, something like that.
Very
I don't
good.
know if I answered.
Yes, and so sticking with, along those lines, how will you select your dissolution media, or what will be the dissolution media? Is it traditional, or in this case for permetro, you're using biorelevant media?
So I think it really depends on the drug. So for example, if you know for example if we use traditional method using some traditional medium, and then we have pretty good results, and for certain drug, especially for certain drug with low BE risk, and I think that's fine. However, for certain group of drug, we already know based on the maybe the biochemical parameters, maybe those kind of drug are high risk for BE results. So for example, for gefitinib, we already know it's a BCS2 weakly basic drug, and its solubility is really pH dependent. So for this kind of drug, if we use traditional dissolution medium, then the pH gonna be a constant. So we wanna use the biorelevant medium to mimic the environment both in the stomach and in a small intestine. So that's why we use a two-stage dissolution method. So we use the simulation solution for the stomach, and then we use the biorelevant simulation solution for the small intestine. So these are two separate parts. And then we will use pumps to really stimulate the movement process by pumping the solution from this cup to the second cup. So that really depends on your drug, I think. So if the drug is highly have high risk for BE, and the absorption is really depending on the physiological environment. So we will maybe try the biorelevant dissolution media.
And is there any separate analytical method development required, or can you just use sort of a traditional HPLC or UV method?
Yeah, I think usually for most of the cases, HPLC method is enough.
Excellent, thank you. And we have another question here. Is the flow from dissolution vessel through the permeation cell a continuous process? And the answer is yes, it is a continuous flow there from the dissolution vessel. And Professor DeCagno, do you want to, we've got a number of questions here. Would you like to take your pick?
Well, maybe just as for custom, the first one that came out, can artificial skin accepted by regulatory authorities? I think this question is crucial. When it comes to regulatory authorities, we are still waiting for new modern legislation. For what this stands at the moment, we have both in Europe and US regulation that allow this so-called bio waivers. That is more for generic compounds. For oral drug delivery, BCS class 1. So in case of, this is already under regulatory approval. So you can apply through the proper channel to have approval without animal testing if you prove that your in vitro testing is solid enough for this class of drugs. All the methods we show now are methods that should accelerate the drug development. So will allow you to reach a prototype and a final formulation, which will be up to standards when you go in vivo, so to reduce the risk. I think with the time given, we are going to come to regulatory approval where this very system will be inside both the European and US pharmacopoeia and hopefully Chinese pharmacopoeia. We have to build science on this. Science is going faster. We have a lot of publications showing the reliability of this method, so it will come soon. For what I can say on artificial skin barrier, I know that Fabio is working on a permeability skin barrier, which is showing very promising results. We cannot, at the moment, we cannot really discuss this yet, but maybe in the next Logan webinar, we will have the chance to also present results of this very promising new barrier for skin studies.
Excellent, thank you. Dr. Lin, please feel free to jump in. We still have a number of questions and we have about 5 more minutes.
Maybe,
right,
oh, yeah, maybe I have just another one, but I can take it later.
Yes.
Kate.
yeah, you
Go
can
ahead.
go ahead.
I just read an interesting one because there is a question about the ratio between surface area and volume ratio, which is often considered crucial for this dissolution permeation studies. Maybe Lynn can comment on this one. It's about how comparable is the surface area volume ratio. For what I know, the system you're utilizing has a very nice correlation within the human condition. But yeah, maybe Dr. Lin is more expert than me.
Surface volume ratio. Yeah, I think I don't think we really like consider this in our research. So we just pick up a appropriate dissolution method based on this drug. So we just pick USP2 dissolution method. And however, we didn't really compare whether, USP1 or USP2 really, which one is better for this drug. So we just pick one.
That makes sense.
Which function, yes.
Yep.
So I can answer a question that is specific for me. So do we have experience or trial in the lab to analysis clopidogrel or atorvastatin drug with permetrol? Is it possible to predict BE with both active ingredients? And if possible, is the result of predict BE of clopidogrel or atorvastatin will be similar or almost same with in vivo test? So actually, to be honest, we just did some lab work to analyze clopidogrel to predict the food effect for clopidogrel. However, we didn't do any work for atorvastatin. However, regarding the BE results for both active ingredients, we also look forward to our results. And we have to say, what's the potential of food effect for this drug based on the results. Yeah, so it's the results of predict BE of clopidogrel or atorvastatin will be similar or almost the same with in vivo tests. That really depends. And that really depends on, how your dissolution experiment were designed and how you find the most optimal dissolution condition based on maybe based on your PBBM model. So that's really depend on your dissolution condition. Yeah, thank you.
Thank you. And maybe Dr. DeCagno, we'll let you finish with one more question.
Yes.
And then I'll conclude after that.
Shall I select from the
Please,
one?
if you don't mind. Thank you.
Well, it's very, I think it's again about regulatory. It's about the permeatral, but it is possible for BCS class 2 and 4 for molecules to avoid pivotal BA by permeatral data. I think I touched it before. For this class of drug, it's still not allowed to have an approval of drugs if you don't go for in vivo testing, but it will probably come. What you can do, and this is the point of this method, is always to reduce the risk to get in the best formulation, to go to a minimum number of in vivo studies, which actually just have to confirm your data for the approval. So for now, unless we're talking about BCS class 1, so very soluble and permeable, unfortunately, also the generics compounds need to kind of wait for this. The BCS class 2 and 4 are still not allowed for bio waivers, but hopefully it will come soon.
Yes. Very good. Well, ladies and gentlemen, we are out of time, and I'll conclude the webinar. And I want to thank you very much one more time Professor DeCanio and Dr. Lin for putting all of your time and effort into this. I think it was very, very helpful. This is our first webinar we've done together. In fact, this is the first one we've done here at Logan. And I hope we have many more down the road. It was really a pleasure working with you. And there were many questions that were asked that we didn't get to. So I think we had a very active audience, which was great. And so look at you, the audience members. We'll do our very best to collect all of these questions, and we'll assign them to the presenters today, and we'll try and get you those answers as quickly as we can. And if you have any more questions down the road, feel free to reach out to us directly at Logan or through your local Logan distributor or manufacturer's rep. And everybody, again, thank you for your time today, and we'll see you at the next webinar. Thank you, everybody. Bye-bye.
Thank you very much. Thank you, Dr. Lin.